Students, I hope you're doing well. I am Honey Rachel. Welcome to this session. I teach biotechnology in K.B.N college. In this session, we'll try to learn something interesting. I hope you all might have watched this kind of testing kit, right? It's called rapid testing kit. Not only pregnant ladies use these, but we use the rapid testing kit for several tests like COVID-19, malaria, and not only these two, we also use these rapid test kits for hepatitis diagnosis. This is a simple kit in which we put sample, mate, blood, urine, or saliva, and the sample is put through the sample region and that sample is processed. The kit identifies if there is any antigen present in the sample. If the antigen is present, then a test band appears in color. If there is no antigen, then there won't be the color change in test band. In either cases, positive or negative, the control band, C band, will always appear colored. The control band will help us understand whether the kit is working or not. So if the band appears color, what does it mean? Yes, you're right, the test is positive. If the test band appears no color, then what does it mean? Yes, you're right, the test is negative. Let us understand what's inside this kit so that it helps us understand or diagnose several diseases. Shall we? Yes. Please watch this video. This will take us inside the rapid test kit. What's inside? Here is the sample pad and here is the nitrocellulose membrane. The sample is taken and it is put in through the sample well, and the sample, it flows through the testing kit and there it interacts with the antibodies. This is test band and this is control band. As the interaction is done, the test line appears in color and the result is positive. And this is case number 2. What if the sample does not contain any virus? What happens is the flow happens in either cases, but the test line stays untouched. Whereas the control line interacts and we get control in color. And that shows us the result is negative. You see, the most important molecule that is present in the rapid test kit is an antibody. And this is not any antibody, but it is monoclonal antibody. In this session, let us try to understand more about monoclonal antibody, what they are, how are they made, shall we? As you have observed, the antibody which is present in test kit is interacting with only one specific antigen. Yes, one specific antigen. For example, in rapid test kit of malaria, the monoclonal antibody that is present in the test kit will interact only with malarial antigen, not with any other antigen, even if present. So, a lab-grown antibody which has only one antigenic determinant. See, it interacts with only one antigen, not with any other, because the site is very complementary to only one antigen. Such antibody which has only one antigenic determinant and which is made in the laboratory, we call it monoclonal antibody. This monoclonal antibody has several applications. As one application we talked about is diagnosing different diseases like malaria, COVID-19, Not only diagnosis, The monoclonal antibody can be used to treat several diseases. Not only that, it can be used as a wonderful research tool to diagnose and treat cancers. So, in research, in diagnosis, in treatment, wonderful, right? Let us understand more about these monoclonal antibodies. These monoclonal antibodies, from their first letters, they can also be called MABs, MABs. Yes. Examples of MABs are trastuzumab, gentumab. The names are very special and very unique, following all the procedure of nomenclature. And if you have observed, in both the names, both the examples trastuzumab and gentumab, there is mab at the ending, which helps us understand it's a monoclonal antibody. The names are given depending upon their function as well. Look at here, metastatic breast cancer. For this purpose, trastuzumab is used, and acute myeloid leukemia. For this purpose, gentumab is used. So these two are specially made in the lab, which can interact with breast cancer and leukemia, blood cancer. Not only that, the monoclonal antibody can be linked with a drug which can be delivered appropriately by the monoclonal antibody. And it can target certain antigens because it has antigen binding site. Because of these features, because of these wonderful characteristics of monoclonal antibodies, they are used in several purposes. Research, diagnosis and treatment, as we have discussed earlier. It was Milstein and his associates in 1970s who discovered this monoclonal antibody, who came up with this idea of using monoclonal antibodies for several purposes. He was the one who first cultured and purified monoclonal antibodies. The production, how can we create these? Yes, they can be created. We can create one particular monoclonal antibody, like we can create trastuzumab in the laboratory for the purpose that it's made for. And this process of making monoclonal antibody, we call it hybridoma technology. Shall I repeat? It's hybridoma technology. Let us understand more about this hybridoma technology so that we can make MABs in the lab. It's a simple process. In this simple process, we need two different kinds of cells. Why two? Let us understand them. First, we need cells which can create antibodies. In our body, we have special cells which release antibodies. The cells which release bullets, yes, these are antigen fighting bullets called antibodies. Who release these? In our body, we have B lymphocytes or B cells which are specialized only for synthesizing antibodies. For example, if we are attacked with COVID-19, B cells understands the COVID-19 antigen and creates an antibody which is quite attachable, shows quite affinity to that particular antigen, COVID-19. Yes, it's B cells who create antibodies in our body. So we need that cells for sure. We need another kind of cells as well. Why? B cells are short-lived. They have particular life cycle and then they'll die. Even we have B cells which can release antibodies of cancer. After few days they will die, right? But if they die, we cannot acquire those particular B cells frequently. That's an painful process for the patient, even for the animal. For this purpose, what we do is we take myeloma cells. You know myeloma cells, these are cells which cannot die, but these can undergo several divisions. They are basically tumor cells which can be obtained from the bone marrow. So the cells which will not die, the cells will produce antibodies. These two together will be a great combination to give us monoclonal antibodies. So for these two, what we'll do first is we will immunize for the first step. In the first step, we will immunize a rat with certain antigen for which we want antibody. If we want antibody, monoclonal antibody for COVID-19, we will immunize the rat with COVID-19. If we want malarial antibody, we will immunize rat with malarial parasite, malarial antigen. So that's the first step, immunization of rat with antigen. After immunizing, the rat slowly starts developing monoclonal antibodies for that particular antigen. So after that, after immunization, we will extract spleen from the rat. In spleen, we have B cells. So from the spleen, we will extract B cells. We have B cells ready. Now we want myeloma cells. Where can we get them from? As I said earlier, we can get myeloma cells from the bone marrow. After extracting these two cells, we are ready to go. Now we want a hybrid of these two. How can we get hybrid of these two? Is by fusing these two together. How can we fuse two cells together? It is possible. It is possible through a chemical called PEG, polyethylene glycol. When we put these two types of cells in PEG medium, they slowly fuse together and form hybridomas. And as we get hybridomas, we will screen them, because while fusing the cells together in PEG medium, we not only get hybridomas, but we might get combinations of B cells, combinations of myeloma cells, and we might get hybridomas as well. But out of all those combinations of cells, we want only hybridomas, of a perfect fusion of B and myeloma cells. So for that, we will take hybridomas into a special medium called HAT medium. What is this HAT medium? The HAT medium contains three essential elements: hypoxanthine, aminopterin, and thymidine. This HAT medium will allow only hybridomas to grow, and the rest of the cells will slowly die. Why? The HAT medium is favorable only for hybridomas to grow. After first selection, that is growing of hybridomas in HAT medium, we will take those hybridomas which are cultured properly, and we will send them for a screening. Whether they are producing proper antibodies or not, that should be checked, right? So for that purpose, we'll send them for screening, and in the screening step, we will allow the hybridomas to interact with the antigens. And when the hybridoma interact with antigen, it will try to release antibodies. And with those antibodies, we will assure that hybridomas are functioning properly. Further, we might screen again and preserve the hybridomas properly. And in the last step, we can use the hybridomas for culturing large amount of monoclonal antibodies. And the monoclonal antibodies can be purified, preserved, used for research, for treatment, and for diagnosis as well. That's how the hybridoma technology helps us make monoclonal antibodies and do research. That's how MABs are made. And I hope this is the flow. This is the flow of hybridoma technology, where we first isolate B cells, isolate myeloma cells, and we let them go fusion. We call it somatic fusion because both cells are somatic cells. And after fusion, the hybridomas are selected, and then selection is done in HAT medium, and then we do screening, and then we harvest the monoclonal antibodies.